Welcome to the Pathology AI Research Roundup for April 15th, 2025. I'm your host, Alex. On today's episode, we're diving into some fascinating new applications of deep learning in human histopathology. It's amazing how quickly this field moves, isn't it? We'll be looking at two key papers that caught my eye recently, exploring how AI is helping us analyze tissue images in new ways. So let's get started. So the first paper we'll look at today is titled, Identifying Regions of Interest in Whole Slide Images of Renal Cell Carcinoma. This one's by Mohammed Lamine Benamar and colleagues, published very recently on ARCS14, April 9th, 2025. The main goal here, as you might imagine, is tackling the sheer amount of information in whole slide images, or WSIs, specifically for kidney cancer, renal cell carcinoma, or RCC. Manually finding the important bits, the regions of interest, is incredibly time-consuming for pathologists. So this team developed a fully automated system to pinpoint these ROIs. Their method is pretty clever. They start by dividing the huge WSI into smaller patches. Then they use some neat pre-processing involving color transformations, focusing on violet and hematoxylin channels to highlight vessels and nuclei, and they even filter out patches with low cellularity. For the core analysis, they used an advanced texture descriptor called Dominant Rotated Local Binary Pattern, or DRLBP. What I mean is, it's designed to capture structural information even when tissues are rotated differently in the image. They extracted these DRLBP features from the key color channels and then compared different classifiers. They tested traditional machine learning like SVM, random forest, and KNN against deep learning, specifically transfer learning using pre-trained models like VGG16 and RESNET50. The results were quite impressive, high accuracy across the board. Interestingly, the support vector machine, or SVM, combined with their DRLBP features, actually achieved the highest precision at just over 99%. The fine-tuned RESNET50 model was very close behind at 98.5% precision. I found it fascinating that a well-engineered feature extraction method with traditional ML could slightly outperform a deep learning approach here, likely because it's very tuned to the specific texture features needed and maybe worked better with their dataset size of about 1,800 patches. This really underscores the potential for automated systems to significantly help pathologists by quickly highlighting relevant areas in these massive kidney cancer slides, saving time and potentially improving diagnostic consistency. Moving on to our next study. This one's called Teaching Pathology Foundation Models to Accurately Predict Gene Expression. Teaching Pathology Foundation Models to Accurately Predict Gene Expression with Parameter-Efficient Knowledge Transfer. This is by Shepan and collaborators, also fresh off the press on ARCS14, April 9th, 2025. Now this paper tackles a different, but equally challenging problem, trying to predict gene expression directly from standard pathology images. You're probably wondering, why do that? Well, gene expression profiling gives crucial biological insights, but methods like spatial transcriptomics are expensive and not routine. Pathology slides, however, are everywhere. The challenge is that image models, even big foundation models, are great at seeing morphology, but often struggle to infer the underlying molecular signals. There's a sort of domain gap. And fully fine-tuning these huge models to bridge that gap takes massive computational resources. So the authors propose a new framework called PEKA, which stands for Parameter Efficient Knowledge Transfer. The core idea is to efficiently transfer knowledge from a teacher model trained on gene expression data, they used scFoundation, to the student pathology image model. They do this using a technique called block-affine adaptation, a type of parameter-efficient fine-tuning, or PEFT, which only modifies about 5% of the image model's parameters. Smart, right? The training involves two main loss functions. One for knowledge distillation, basically making the image model's predictions mimic the gene expression models, and another for structure alignment, ensuring the relationships between samples in the gene expression space are preserved in the image model's feature space. They tested PEKA on several spatial transcriptomics datasets covering breast, kidney, liver, and lung cancer, over 200,000 image tiles in total. The results? PEKA significantly outperformed baseline foundation models like RESNET50, CTranspath, UNI, and Optimus 0, boosting the prediction accuracy, measured by Pearson correlation, by at least 5 to 7% across all datasets. It also beat other parameter-efficient methods like LoRa. This suggests PEKA is really effective at bridging that modality gap and letting image models understand molecular information better, all while being computationally efficient. I think this is really significant. It opens doors to potentially extracting rich molecular data from routine slides, which could be huge for research, and maybe even clinical applications down the line. So, wrapping things up, today we saw two really interesting approaches using AI in pathology. One focused on automating the visual task of finding cancer regions efficiently, while the other worked on bridging the gap between images and molecular data, also efficiently. It really highlights how AI, both deep learning and clever traditional methods, is being refined to extract much deeper and more useful information from histopathology slides. I'm personally excited to see where this research leads next. That wraps up our research review for this episode. Thanks for tuning in to the Pathology